So I'm Jackie. I work in the Leach Lab in the Biomedical Engineering Department, and today I'm going to share my work on mesenchymal stem cell spheroids. So how we apply these adult stem cells is with bone defects. Now, my little brother is part of the 6.2 million people in the United States who break a bone each year. He is also part of the 5 to 10% whose bones did not heal after fixation. Now, you might be looking at his x-rays here and wondering where specifically that fracture is, if you're unfamiliar with x-rays. So I've put a box around it here, and a box around what appears to be a very large screw in his hand after a surgery. And this is the current gold standard of treatment for these type of non-healing fractures. Now, what you do is, looking at the middle image there, you scrape bone from another area on the body, and then take that bone and put it to the non-union site. It's incredibly painful, as my brother has told me, and there's a limited amount of supply that can be taken that doesn't then damage a secondary bone. So what we're looking to do with our research is to eliminate that secondary surgery and have an increased supply using mesenchymal stem cells. So these stem cells, which I'll refer to as MSCs, we don't just take them in the typical monolayer culture. We aggregate these cells into what we call spheroids. And I'll be using this term for the MSC aggregates throughout the rest of my talk. I've also included an image of the tissue engineering triad for those of us who aren't in tissue engineering, to help kind of walk you through why each of these steps are important for developing new bone tissue. So it incorporates cells, regulatory signals, and biomaterials in order to regenerate specific tissues. Now, using spheroids, they're particularly beneficial for several reasons. For this talk, the most important part is that they can become bone, and that aggregation into spheroid form does not inhibit its ability to become bone. But we want to enhance that. We want to take it one step further. How can we use adult stem cells in a more targeted, directed manner? We can use instructive cues, those regulatory signals from that tissue engineering triad. Now, specifically, using bone morphogenic protein 2, BMP2, is already done by orthopedic surgeons. So why would we want to then add it in research here in an academic setting? Well, because how it's currently used, you see ectopic bone formation and a great deal of inflammation, causing pain to the patient. An additional instructive cue that's commonly used is hydroxyapatite. Now, this is the main mineral component in bone. It's typically used in hard scaffolds, but what if we were to use it differently and incorporate BMP2 as well? And that's exactly what we're doing. We're using HA in nanoparticle form to direct the delivery of BMP2 in a more targeted manner. Specifically, we're incorporating BMP2 HA into the spheroids directly. So that BMP2 HA are our regulatory signals, the MSC spheroids are our cells, and then they will be entrapped in alginate, a natural polymer, to then regenerate bone. Now, this is a twofold hypothesis, the first being that of induction. So can it become bone from the stem-like cell? How we're going to do that is to take the HA, load the BMP2 onto it, incorporate it into the cell suspension, and form spheroids. Now, here we don't use any other form other than osteogenic media for 14 days. And we're going to then evaluate the osteogenic phenotype, or bone phenotype. Additionally, what's important is, can it keep that phenotype over time? Or are the cells going to revert back to their multipotent potential? So to help you remember that, I've included little graphics here so that induction means becoming bone and retention means locking in that phenotype. So the first place that we started was looking at, well, can HA be incorporated into spheroids, and will we still see that beneficial component of spheroids that I alluded to in the introduction? And if so, how much? We started with 4 different concentrations of HA, and what we see, just looking at it gross morphology-wise, as well as quantifying the diameter of these spheroids, is there is an increase and a change in size with an increase of HA concentration. Now in our lab, we indicate significance with letters. So if it has a different letter, that means that they are statistically different. So then when looking at the DNA content where it says NS, that means it is not statistically significantly different. And that this is important because that means we have the same number of cells in each of these spheroids, and the size is being changed with the HA incorporation. Now you can tell from just looking at these images here that one concentration specifically was too much for the spheroids to retain their aggregates. So we eliminated that concentration moving forward with our studies. When looking at the spheroids cross-sectionally, what we noticed were larger voids than the 100 nanometers of our commercially available hydroxyapatite. So then we investigated what these nanoparticles look like using SEM, as well as quantifying their diameter with a zetasizer. What we see is that there is in fact clumping of the HA nanoparticles, as well as with increased concentration, there is increased clumping. Next, we moved on to, can we load our growth factor that will help instruct bone morphogenic formation onto the HA? And the answer here is yes. So we fluorescently labeled our BMP2 and loaded it onto our 3 concentrations of hydroxyapatite. We then looked at the fluorescence under a microscope and quantified it. Our comparison here in the middle column is hydroxyapatite with soluble BMP2. And this is how BMP is typically delivered. So we wanted to compare our results of the incorporated instructive delivery of BMP2 to what is currently done in the field. In terms of quality control, we lastly wanted to see how does this incorporation of BMP2 onto HA into the MSC spheroids affect their viability. You can see in the live dead images at the bottom where green denotes that these spheroids are in fact alive, that they are with both the concentration and with just OMHA, which means osteogenic media with just hydroxyapatite, and OMHA sol BMP, which denotes the osteogenic media with HA and soluble BMP. Again, importantly, we don't see a difference in the DNA content, meaning we still have the same number of cells, and we don't see a difference in their metabolic activity. The line graph on the left denotes that there is a decrease in spheroid size over time, no matter the incorporated components, and this is indicative of spheroid behavior in culture. Finally, when looking at the ability of these instructive cues to initiate or induce that osteogenic bone phenotype, we looked at alkaline phosphatase activity, or ALP. This is a mid to late marker for osteogenesis, and what we see from day 7 to day 14 is an increase using our BMP2 loaded HA in specifically the 0.5 to 1.0 milligrams per mil. Next, we looked at gene expression, specifically RUNX2, which is considered the master transcription factor for osteogenesis. What we see is a much higher gene expression for our BMP2 loaded onto HA, incorporated into these spheroids at the 0.1 milligram per mil concentration. Next, moving on to retention, we took a slightly different method in terms of measuring the osteogenic potential. We looked at calcium deposition, which is a late marker for osteogenesis, since we wanted to see if they could maintain their ability to be bone cell-like. The red line denotes how the calcium deposition was expressed on the very first day when we immediately took them out of that osteogenic media. On days 1 and 5, they're now in growth media, and we're looking to see if they revert back to their stem cell-like phenotype. We see that in all groups, they do revert back. However, in the BMP2 loaded onto HA and then incorporated into the spheroids, specifically at the 0.1 and 1.0 milligram per mil concentrations, a recovery in that osteogenic phenotype is seen from day 1 to day 5, as denoted here by the arrows. So in conclusion, what this study has shown us is that we can in fact load BMP2 onto HA nanoparticles, incorporate those HA nanoparticles into mesenchymal stem cell spheroids. However, when inducing the osteogenic phenotype, we did not see the type of tunability that we were really looking for, which can be done using other soluble methods put into the osteogenic media in other labs. However, in retention, we did see a restorative behavior, the ability for BMP2 to be incorporated onto HA, then put into the MSC spheroids, and help maintain that osteogenic phenotype when bone cues are removed from the media. And with that, I would like to thank my lab, acknowledge my funding sources, and take any questions you might have at this time.